Kate, this is David. It's good to see you.
Hey, David, it is awesome to see you too. And you guys might notice a little bit of flash photography in the background. That's an experiment that we've set up to run overnight, so don't let that surprise you.
Great. Somehow this is familiar territory to me. So let me just first welcome all of you here to Berg Hall at Stanford University School of Medicine. I'm David Relman. I'm a professor in the departments of medicine and of microbiology and immunology here at Stanford. It's my great pleasure to introduce Kate Rubins, NASA astronaut, current resident aboard the International Space Station, and former Stanford PhD doctoral student and Stanford alumna, for whom I had the honor of serving as thesis co-advisor along with Pat Brown from the Department of Biochemistry. Kate, so the first thing I have to say...
Hey, it is great to talk to all of you guys.
Kate, I have to say this because how often does one get to, but greetings from Earth. And second, of course...
That is a good one. I'll say this because I don't get a chance to say it too often either, but greetings from space to all of you.
And of course, the second most important comment, how are you and your microbiota?
We are doing very good up here. I did have to ask the microbial analysis folks if I could use a few extra of their swabs that they've sent to space station because it's so fascinating up here, and I probably would use every single swab that they sent and then run them out of swabs. So I have to moderate my excitement about microbiology just a little bit up here.
Okay, well, we can talk about that. Let me just explain the plan for the next 28 minutes or so, while Kate travels about 1/3 of the way around the globe, is I'll give you a brief intro to her career, very brief, open with a question or two, and then give Kate a chance to say a little bit about her current work. And then I want to spend as much time as we can with questions and answers from all of you. We have 2 roving microphones. She will hear you. As you know, there is a little bit of a time delay, much like here in your car in Silicon Valley. And I'll ask that you identify yourself before asking your question. Also, just to make clear, Kate cannot see you, but she can hear you, and we can see you. So just briefly, Kate was born in Connecticut. She was raised actually here in Napa. And then received her bachelor's degree from the University of California at San Diego. It was 1999. She then came here to Stanford, and soon after her matriculation in the graduate program here, she approached me and Pat Brown with interest in the work that we were doing to understand how humans and other mammalian hosts respond to systemic infection. This was using host genome-wide transcript profiling. Kate was immediately drawn to the problem of unraveling mechanisms that underlie fulminant viral disease and pursued collaborations that we had set up with the CDC and the US Army to study smallpox and Ebola viruses, live viruses. Kate then spent a significant fraction of her graduate student days literally working in a spacesuit with these 2 viruses in BL4 facilities. She developed the first model of smallpox. She elucidated molecular mechanisms for the first time about how smallpox causes disease, and she contributed a number of important findings and publications to the scientific literature. She was fearless, she was curious, persistent, upbeat, and amazingly dedicated to science. I would get phone calls from her from Atlanta, much like this, as she started to say, describing some unexpected challenges that had come up in the middle of the night during an all-night experiment in a BL4 suite with smallpox virus. And then she would tell me about how she and our collaborator, Lisa Hensley, had concocted on the fly some makeshift scheme to solve a problem that had taken place. gave me the willies, needless to say, as her mentor. She received her PhD in 2006. She then became a Whitehead Fellow at MIT. She organized an independent research program there, had her own lab. She was studying monkeypox and host interactions. And then, 2 years later, NASA issued a call for applications to the astronaut program. Kate called me and revealed a childhood dream of hers that she had not previously about her desire to travel into space, and asked if I would write her a letter of recommendation. It was one of the easiest letters I ever wrote. Kate was then selected in July of 2009 from thousands of applicants as one of 14 members of NASA Astronaut Group 20. And after a grueling training period at NASA, was then picked last year as a flight engineer for this ISS Expedition 4849. She became the 20th woman to travel into space when she launched this past July 6 on Soyuz MS-01. So, Kate, I gather you have even less bench space now than you did over here in CCSR.
I actually, we do have little mini work areas, and the first thing I did was clear off, there was some electronics debris on there that previous crew had left. I cleared off the lab bench, and I pulled out my pipettes, my tips, and my tubes that I have packed in my little small 1 kilogram allocation, and I've got all of that set up right next to my sleeping station. So whenever the urge strikes, I can go do an experiment or two.
Sounds familiar to me. So let me just ask you to start. What would you say are the most important questions that you and NASA are asking in the life sciences up on the station?
I think that's a really great question, and I would say the two things that are the most fascinating up here are exactly what you said, the microbial life on board space station. And I thought about this a lot before I launched. I think that we're on Earth now discovering microbiomes everywhere. We're a little bit, I would say, in the computer age, maybe in the mid-'80s, in terms of our ability to completely miniaturize, to use sequencing to around us. And I started thinking about the space station. We have a microbial community up here that the space station's been orbiting the Earth for 16 years, and it launches not sterile, but mostly clean. It's clean room equipment and hardware. That you then put humans and all their associated flora into the space station, and you have this giant experiment of this laboratory orbiting the Earth. It's been exposed to radiation. We have levels of radiation because we're a little further outside the Earth's magnetic field. We get exposed to some fascinating heavy particles up here. It's a mix that we can't simulate on Earth. And then, of course, these microbes are finding their niche in their environmental habitat up here, and they're growing and they're changing, and I think we're going to be able to look at that in a pretty new way with some of the sequencing technologies we just demonstrated on this increment. The second thing that I've been incredibly fascinated by is fluids up here. So we're in free fall. I'm in free fall right now. I'm falling around the planet, and you sort of know this in principle, and then when you actually start to float, and you're floating a little bit independent of the structure around you, it gets demonstrated really on a daily basis up here. So I can float completely in free fall if I stabilize myself pretty well, and that does fascinating things to fluids. So fluids are not subject to the force of gravity anymore. They don't sit down at the bottom of any vessel. They ball up. Bubbles form in systems, microbubbles, and all of biology is related to fluids. So I think the investigation biologically of what's going on, everything from a microliter up to a human being, is incredibly fascinating up here, and we've just started to scratch the surface of this.
So I will note that you're one of the few people who would refer to their own whole body irradiation as fascinating. But let me just ask you, I know it's early, any interesting, unusual, unexpected findings from what you've been doing?
Well, we're hopefully submitting a paper on Tuesday about some of the first sequencing from space.
Yes, I've read
I
part
don't
of it
want
already.
to publish before. Yes, I don't want to publish before we publish, but stay tuned. It's fascinating. I think the bottom line is it worked incredibly well in low Earth orbit. We weren't necessarily expecting that, and we are going to venture as a species beyond low Earth orbit. This is going to happen. I'm really hoping going to happen in our lifetimes. And as we do this, one of the big driving forces is to look for life elsewhere. I think the way that we're going to do that is sequencing technology. And so I'm just feeling incredibly privileged to be able to be on space station dawn at the very beginning of this molecular age beyond our planet.
So one last question, then I want to open this up. As you look back at your days here at Stanford in graduate school, is there something that you think was particularly important in preparing you for doing what you're doing? Or maybe the converse, that hindered what you now realize you needed to have learned how to do?
I don't think there was any hindrance at all. I think getting the chance to be at Stanford, to do work there, to train under you and Pat and all of the associated mentors, friends, colleagues there, that's what teaches you as a scientist. So what you all are doing at the med school, in materials, in engineering, all of the folks in the different schools there, what you're researching right now, what you're doing, is forming you as a scientist and as a researcher, and I think that was incredibly important. I'm putting that to use every day up here. So I think it's pretty fun to be a scientist in space, but it's also incredibly fun to be a scientist on the Earth. So I hope some days when the experiments get a little grinding, that you guys do think about how incredibly lucky we all are to just be able to observe the world around us.
Thanks. We do hope that you will become a scientist on Earth again at some point. So we're going to open this up for questions. Again, if you could first raise your hand, I'll try to be sort of equal in my dispersal of the microphone, and if you could just identify yourself. So maybe here in the center. Is there a microphone we could bring over here? Oh, they're up at you have to come up, sorry, to the stand. Kate, just 1 minute while someone comes up here.
Good day, Kate. I'm Zina.
Sure, no problem.
PhD student in immunology here. My question is about detecting life on other planets. And I'm hearing about life chip, which is an antibody array with antibodies against various pieces of DNA, RNA, protein, and small molecules. And my question is, what do you think is the best proxy of life that we could search for?
I think that's a great question, and I think any instrument that we send to another planet, future planetary missions, crewed missions outside low Earth orbit, are definitely going to have a suite of tools that's going to be trying to detect life. I think antibody detection, using antibodies to detect, is fascinating. I think we're definitely going to have some sort of detection method for nucleic acid. The thing is, we don't exactly know what we're looking for. So nucleic acids off the Earth may be different than what we're looking for on the Earth. And so I think actually employing a variety of tools of all different, basically everything in our arsenal that we've got, we're going to take a look at that, and we're going to see as chemical structures are changed and modified, potentially how can we use our Earth-based tools to look for life elsewhere. So I think it's fascinating ideas. I think if you want to work on developing some payloads, I would bet that NASA would take you up on that.
Okay, question on the other side of the room here.
Hello, Kate. My name is Gustavo. I'm a student at geophysics. And a couple of days ago, there was this big conference on Aero-Astro, and Elon Musk presented this big project on the engineering challenges of colonizing Mars, but he didn't really touch anything about the biology difficulties involved with that. Could you talk a little bit about what you think are the biological challenges of colonization?
That is such a great question, and I think we are driving towards moving to living on Mars. Space Station is an example of where we're taking our first steps for that. So we have an environmental life support on board Space Station. It's what's keeping me alive right now. I'm very grateful for it every day, and it recycles 90% of our water. So it's an almost entirely closed loop system. We don't have to launch water from Earth anymore. We used to do that during shuttle days, but we now recycle our water. So we pull water out of the atmosphere. We take all of the condensation from the atmosphere. We take our coffee in the morning and we turn it into tomorrow's coffee, as we often say up here. We pull every bit of water out of the system and we recycle it. So that, I think, is a proving ground for technology for sustaining life off the planet. We do the same thing with air up here, and so we'll take the water molecules, we'll split that, we'll make oxygen for us to breathe. We need to pump a little oxygen into the system, but we don't need to supply huge tanks of oxygen for the space station. We're making our own atmosphere up here. We scrub the CO2 out of the environment, and so it's really a giant experiment about how you can take a completely closed module, put humans in it, and do all the things that they need to do to survive. I think those are going to be the baby steps for us. It's an engineering and a biological interface for how we're actually going to live off the planet long term.
Drew.
Hey, Kate, this is Andrew Herkewine. I'm a postdoc in Justin Sonnenberg's lab. We're going to go to the gut microbiota. You mentioned the sort of community that's been evolving over time in the space station. I'm curious, in lab we focus on diet. I'm wondering how much dietary fiber and other things you're feeding to your microbiota while you're in the space station.
Well, I'll tell you, without going into too many of the details, digestion definitely changes up here. It's actually, I think, profoundly influenced by microgravity and your internal organs floating. So there are changes up here. I think we're just starting to look at the beginning of those. We do eat a steady diet of space food up here. So it all comes in a little package. It basically looks like astronaut ice cream. It's all freeze-dried, and we just rehydrate it. We do get a fair amount of fruits and vegetables up here, so we are hopefully feeding our bugs pretty well. I know there'll probably be some studies down the road to completely profile the microbiome up here, the digestive microbiome, and I think it's going to be really fascinating. Astronauts are an extremely well-controlled, mostly well-behaved subject population. So we have complete control of environmental factors up here, and I've recently been working with some folks on a food logger app that logs every morsel that goes into my mouth, so we can actually, I think, get some incredibly well-controlled data about space diet and about the microbiome.
And as we know, Kate, there's some great human subjects experiments underway already to address those questions. On the other side of the room.
Hi, thanks for talking with us. My name is Jonathan Herman. I'm a structural biology graduate student, and this is a bit of a silly question, but how does pipetting and transferring small amounts of liquids work in zero gravity?
That is not a silly question. That was my #1 question that I wanted to answer coming up to space. It's a really phenomenal question, because for the last 20 or 30 years of space biology, we've generally assumed that pipetting is not going to work so well up here. I was talking about things floating. You know, we float, liquids float. But I wanted to see, when you start to get less than a mil of fluid, surface tension is really also a dominant variable up here. And could you do pipetting with commercial hardware? Can you just take a lab bench and fly it to space? It's obviously not that easy, but design a special, completely enclosed system. And again, I need to write a paper and I need to publish all of this, but the answer as of a few weeks ago is pipetting works completely fine in space. The volumes are so small, the surface tension is so dominant. If you have held a pipette in your hand for any number of years and have a decent amount of control, you can do an experiment just like you would on the ground.
Over here.
Hello, my name is Jody Lang. I'm a physician anesthesiologist here at Stanford. Pleasure speaking with you. According to Wikipedia, there were 14 people in your class, and I was wondering how'd you get to be one of the two that made it up there?
Well, we all fly in our class at different times, and so I'm about a third of the way in. We've flown part of our class. The rest are assigned or waiting to fly. We have a steady rotation of astronauts that come up to the space station, and we all need a number of years of training. We don't specialize so much anymore. We used to be very specialized at NASA in terms of our pilots and our mission specialists. I would say I'm probably more in the mission specialist category, but they had to teach me how to fly. I fly T-38 jet, trainer jet now. I went to the Navy, soloed at T-6, went to flight school. And then we teach our pilots how to be scientists. So we conduct courses for them on microbiology, cell culture, immunology, rodent research, anything you can imagine. So it takes quite a number of years to do this. And so you can imagine, as we're training all of these classmates, they're progressing through the training program. And you just kind of wait until your number is called and you get the opportunity to fly in space.
Dan.
Hi, Kate. I'm Dan Sprocket. I'm also a graduate student in the Relman lab now, so pleasure to talk to you. You're doing some really sophisticated molecular biology right now, and I was wondering, now that you've had an opportunity to run some experiments, if you weren't limited by funds or space, what's the next generation of questions you'd like to tackle?
Ooh, no funding limits. I like this one. I think that we've just started to do some experiments with cell culture up here. So that actually was a Stanford experiment. We grew cardiomyocytes up here long term. They were up here the entire time that SpaceX 9 was docked, and we sent them back to Stanford. And I think the ability to look at cells long term. We've flown cells before on shuttle. This isn't an entirely new thing. We've flown cells on space station, but we haven't had the setup where we could really do long duration experiments, culture cells, and investigate the cellular biology. I think looking at transcriptional responses, obviously that's near and dear, but the structural biology of the cells, if you plate fibroblasts on Earth, they go to the bottom of a dish and they stick and they spread out all fibroblasts like. What's going to happen to them up here when they don't have a gravity vector, when they're all in free fall? You can do clumping experiments where you seed the culture wells with various types of beads. You could make adherent structures. NASA's putting out a research call to actually start building vascularized tissue chips on orbit. You all should look for that if you're, I don't think it's unlimited funding, but it's a source of funding. And I think just trying to understand what happens to cellular biology and the associated molecular profiles when everything is not constantly influenced by this downward gravity vector that we have all the time on Earth in every Earth experiment.
We have about 3 minutes left, unfortunately. Maybe a question over here?
Hi, Kate. This is Howard Chang, faculty dermatology. It's good to see you in space.
Hey, it's great to see you.
Yeah, wonderful. So you share with us the exciting result about the sequencing in space. And so, of course, you can sequence DNA. Maybe you can talk a little bit about, can we get into understanding the epigenetics in space? And the prior work with the prior mission with astronaut Mark Kelly involved really complicated ways to bring samples back to Earth. What would be the future possibilities?
Yes, we are absolutely working on that. There's going to be follow-up studies to that twin study that you mentioned. I think we can start to do a lot more analysis on board the space station. We're working on a few pieces still to do some things like library prep, but I would say most of the pipetting experiments I've done, I think we've fairly well demonstrated that that's not going to be too difficult to assemble. And we are going to expand. Obviously, we've got a nanopore sequencer up here. It's working for DNA. There's no reason we can't do RNA sequencing. There's no reason we can't do any of the epigenomic profiling, the antibody capture and sequencing type experiments. So I think, we have the possibility to get samples back to the planet. I think we'll continue to use those. We have a lot more analysis tools available to us on the planet, but we also are starting to do a lot more in situ. We're doing it up here, and we're sending the data back. So that is something that doesn't rely on a space vehicle. It doesn't rely on complicated methods. We are very, very good at transmitting data from the space station back to the planet.
Okay, one last quick question.
Hi, my name is Maud David. I'm a staff researcher. I'm a microbial ecologist. And so last question was, so did you already see difference in enrichment with bacteria that are resistant to radiation specifically, like Deinococcus or something like that? Or is it too early yet to talk about that?
Yeah, so we haven't been able to take a look at the complex microbial communities quite yet, like I would like to. So I think there's some sampling that's been underway already, and so those results are going to be coming out as they start to analyze the data. And I think we are going to be looking for radiation-resistant bacteria, but I think we're not necessarily growing radiation-resistant bacteria here. There's radiation is a phenomena, but it's not necessarily a selection pressure. I think the more interesting selection pressures are going to be looking at the iodine that's in our water here to see if we're having any microbial breakthrough in our water recycling system. We've got iodine on the U.S. side and silver on the Russian side. And I think we're just then going to observe the cellular effects of radiation on eukaryotic and prokaryotic cells. If you launch some frozen earth bacteria up here, you could thaw that out and take a look over time. And I think eventually we need something like a long-term chemostat up here, take a look at yeast as an ice system to look at those effects, and just any long-term, well-understood genomic changes.
Great. So Kate, I want to just thank you first for taking time out of a busy schedule, sharing some time with us. Also want to thank you for all that you've done for the nation and for the global communities. It's really wonderful. It's inspiring. Thank you for that. A few other quick thanks. I want to thank NASA, and in particular Christina Koch, who, and her colleagues in the Astronaut Office, who did a great job in putting this together. I know that you and they have been doing a number of these outreach efforts for STEM education and other outreach efforts, and I think that's really a great thing that you guys do as well. So thank you all for that. Also, thank you to the Dean's Office here at the School of Medicine for putting this on, supporting it, for the Alumni Association as well, for their support. Finally, thank you all for packing this room. You can't see, you've got a standing room only audience here, and they're beaming, as am I. So we really appreciate all that you've done for us today, especially. So signing off from Earth. It's been a real pleasure.
Well, I want to thank all of you guys for coming, and I will sign off upside down because I can from the space station.